So this is a good pineal gland. Look at the pineal gland. Pineal is a very small gland that's located just above the superior colliculi of the brain. Superior colliculi of the brain. It is invested in a capsule, as you know, a thin, delicate capsule of pia mater. And although it might not be obvious to you, this capsule sends trabeculae, delicate, discrete trabeculae inwards, and these divide the gland into lobules, and it's not very clear, but that's the story. Also note, all these red areas represent blood vessels, so the pineal is no different from most endocrine organs, being highly vascular. Then now, the parenchyma of the gland is organized pretty much like how all the endocrine organs are organized. Look on that. Clusters of cells, so, or what we call cords. So the cells are organized into cords. And again, note that associated intimately with these cords, numerous capillaries. Right at the tip of the pointer, you will see one. And then now we look at the cells of the parenchyma. So these parenchymatal cells are basically of two types. A, the main cell types, which are these pale ones that we see me pointing at here. These are the so-called pinealocytes. Pinealocytes. So the pinealocytes tend to stain very pale with H and E. Also, there are some other cells that are called glial cells. So there are glial cells there like this one. So the glial cells tend to be spindle-shaped. But of course, for you to see the cytoplasmic extensions, now you need special silver staining and so on to demonstrate those. So the only thing you have to go off is the fact that the pinealocytes are rounded pale cells, whereas the glial cells are sort of spindle-shaped cells. Also, it's very important for you to remember that the pineal gland has a very important function in the so-called circadian rhythm. Circadian rhythm, which is how the body responds to changes in light and darkness, and how that affects now the pineal secretion and hence other hormones of the body, including those reproductive hormones, etc., etc. The real influence of the changes in the light and darkness on this gland is via the optic tract to the superior salivate, to the superior cervical ganglion, and then from there, the superior cervical ganglion sends postganglionic fibers now to the pineal. These fibers travel in the nervus coronary. Not canary, but coronary. So, yeah, the nerve supply to the pineal is sympathetic, and it comes from the superior cervical ganglion. These postganglionic fibers from this ganglion travel in a nerve that's called the nervus coronary. Two eyes at the end. Listen, Luca, two eyes, not one. And then now, when we ask to give the nerve supply, you don't say canary, but coronary. With age, look at this now. With age, look what we find. With age, we find the accumulation of these numerous concretions known as brain sands. Brain sands. Or you can call them the correct name, Porpora arenacea. Porpora arenacea. Porpora arenacea. Yeah, if you can't spell that, just say brain sand. Yeah, SANDS. Brain sands. Now these brain sands are actually accumulation of calcium carbonate and calcium phosphate in the secretory material that comes from this gland. So this occurs with age. Pre-pubertal subjects do not show these, but with age, as you get more and more mature, they accumulate more and more. And carbonate, calcium carbonate / calcium phosphate. Now interestingly, the pineal cells, these pinealocytes, we know that the main secretory product is melatonin. Melatonin. But that is not to say that the gland doesn't produce other substances. So it produces serotonin, it produces dopamine, it produces some adrenaline, and so on. But the main secretory product is the what? Melatonin. Not melanin, but melatonin. Alright, so that's that. In the past, I have seen questions like, what's the radiological significance of these concretions? Radiological significance. Dr. Suja, have any notes to add on the radiological significance of these things?
Yes, sir.
Being a clinician.
It's basically there is a space of confusion which shifts the pineal sands, the brain sand, if that is present, to one side, so that it goes off the midline.
Anything else?
That is basically.
Okay, what about the radiologist using it as a landmark?
That is it. It's in space of lesions, it's a landmark.
Okay. Also, you didn't hear that? No. What happened? Dr. Such not projecting her voice enough? Dr. Such.
No, I projected it to you.
Oh. Okay. What she's saying is if there are any space occupying lesions, this lesion or lesions will displace these concretions. And so radiologically, since they are solid structures, you can see them on the radiograph. So you can sort of use these as some sort of geographical landmark, find a way around. Yes. But try not to be as verbose as we were just now, right? We want 1 or 2 word answers. See if you can synthesize that. Alright guys, so that's that. Let's press on. Go on now. Another pineal. Another pineal. So you have to now be observant, because what we tend to do is sort of put the pineal on the periphery of the field. Put the concretion, sorry, on the periphery. So just to complicate life a little bit, let's do something like that. Something like that, right? And put the point, and have the pointer, so guys, and have the pointer pointing at something all the way over the next side. So we say identify the structure on the distal part of the field to distract him. So you have to be very observant, right? Alright, so that's that. So we leave the endocrine now and we go to